Hi there. In this video, I'll be talking about cancer, which is a breakdown in gene expression control. And this is just part one of a two-part video series. Cancer is a loss of gene expression control. Cancer is also called oncogenesis. And it's really a set of diseases involving evolutionary processes such as mutation, recombination, amplification, that lead to the cells becoming immortal and failing to respond to their environment. So it's really a loss of control of gene expression in that the signals that usually control gene expression are lost. So the characteristics of a cancer cell is that there's uncontrolled growth, mutations in the signal transduction pathway are often involved. There's no apoptosis, that is, no cell death, so they're pretty much immortal cells. And then there's loss of contact inhibition. It has no sensing of cells around it, loss of anchoring to other cells, so it starts moving around, and reduced need for growth factors. That is, it'll grow whether the growth factors are around or not. It becomes much more dangerous when it starts to invade other tissues and interfere with the functions of those tissues. And when it starts to attract a blood supply, which is called angiogenesis, where now it has a larger supply of nutrients. And then finally, the ability to move to distant sites, which is metastasis. Here's another way of looking at the same thing. So they have limitless replicative potential. They keep dividing. They can attract blood vessels to themselves. They don't die, so they become immortal. They are independent of growth signals. And then they have an insensitivity to anti-growth signals, that is, contact inhibition. And then finally, when they become more dangerous, they invade other tissues and then start moving to other sites in the organism. Here are some of the general categories of cancers and the origins of those cancers. So carcinomas are cancers of epithelial cells, and those can be of the skin, lung, breast, prostate, pancreas, and colon. And they come from ectoderm tissue. That is, ectoderm is the surface tissues in the embryo and even at the blastoderm stage. So I'll talk about these in a few minutes. Sarcomas are those of the connective tissues, including bone, cartilage, fat, nerve, and those originate from mesoderm and mesenchyme cells. Lymphomas and leukemias are from blood-forming cells, and those also come from mesoderm and mesenchyme. Germ cell cancers are from endoderm tissue, and those can be testicular or ovarian. And then blastomas are those formed from immature cells, and they're usually in children. And those can be ectoderm, mesoderm, or endoderm in origin. In some ways, cancer cells start taking on a character that is similar to their early development stages. So they are more or less undifferentiated, although some are more differentiated than others. So this relates back to their origins in the blastula and gastrula stages. So early in development, you have several different types of tissues that are formed. The ectoderm, which essentially develops into epidermal tissue, and that includes the outer layers of the tissues of the skin, in the gut, and in other types of tissues as well. The endoderm would be internal tissues, eventually develop into internal organs and internal tissues. And the mesoderm is formed through interactions of those two types. So interactions between the ectoderm and endoderm start to form the mesoderm tissues. And so you can see that here, where you start out with a small bit of tissue and then that becomes larger and larger, but is sort of intermediate between the ectoderm tissue, ectoderm cells, and the endoderm cells. So, for example, skin cancers typically would revert back to more of their characters as ectoderm rather than any other type of tissue. So they would tend to do that. Most of those actually stay put, so they don't metastasize readily. However, mesodermal tissue, as well as mesenchyme, which is a type of mesodermal tissue, those influence other cells. So you can see here this mesenchyme in the blastula those cells start migrating, and they start influencing other cells, including invagination of the early gastrula or gut, and eventually do pull that tissue or influence it to move up to form the gut tissue. Now, those tend to move around, and so many of the cancers that are formed from mesoderm and mesenchyme, some of those metastasize quite early in the progression of the cancer. This figure indicates where some of those tissues eventually wind up in the organism and how they develop. So from the ectoderm, you have skin, neurons, some pigment cells, although some pigment cells, like the melanocytes, are actually more mesodermal than ectodermal. And then the mesoderm is muscle, bones, blood cells, things like that. Those tend to move quite a bit, so those will metastasize quite early, especially these melanocytes. If you have a melanoma, those tend to start metastasizing very early in the progression of that disease, which makes them more dangerous. And then endoderm tissue, some pancreatic cells, some thyroid cells, and lung tissue, at least some of those. And then the germ cells, including testicular and ovarian cancers. Just to illustrate this in one cell type, up here, and these are all blood-related cells, and so you have these multipotential hemopoietic stem cells. So this is a stem cell, and often when you have a cancer, it will revert back to sometimes these very early stages. And so it'll act more like a stem cell than a differentiated cell. As you get out here into more differentiated cells, then those don't typically end up as a cancer, although in a few cases they can. So the hemopoietic cells will divide, and they'll form either a myeloid progenitor or a lymphoid progenitor cell. And again, those can become cancerous at that stage, such that anything on this side, these myeloid ones, those are characterized as the leukemias. And then these ones over here would be the lymphomas coming from the lymphoid progenitor. Anyway, so these can then differentiate into keratinocytes that form the blood clotting proteins, and then erythrocytes, of course, red blood cells, mast cells, myeloblasts, and these ones here are typical white blood cells, and so you can have leukemias where you have these somewhere up here becoming cancerous, becoming more numerous. So you have a high white blood cell counts, which are one sign of leukemias, that you have a propensity for a certain type of white blood cell that has become a cancer. Whereas on this side, you would have cancers that are primarily lymphomas of the T and B cells. So if you look at the frequency of certain cancers, this is in the world, Breast cancers far outnumber any other type of cancer. Lung is second, mainly caused by smoking, or in many cases by smoking. Colorectal, melanomas, nervous system, etc., going down the line. So breast cancer is of high prevalence, and certainly, women can get breast cancer, but men can also get breast cancer. And normally, it's more fatal in men because they don't recognize it as being a breast cancer as early as women might. Because breast cancer is so prevalent, then if you look at all women, then about 12% of women will experience some sort of breast cancer during their life, and 88% will not. The large majority of those, 73%, are new mutations just in that person, so they're not inherited diseases. But about 27% are familial cancers. 20% of those are what are called low penetrance. That is, not everyone who has those particular genes will develop breast cancer, but many will. 5 to 7% in here have what are called high penetrance in that if they have those particular genes and those mutations, then they have a much higher likelihood of developing breast cancer. And this is what happened to Angelina Jolie, right? Her mother had died of ovarian cancer and had breast cancer too. And so as a way to, Angelina Jolie was then tested genetically and found to have the same mutation and decided to have a double mastectomy to try to avoid getting that cancer, where she had a very, very high probability of developing cancer. While the incidence of breast cancer is quite high, it does not have the highest death rate. So the annual mortality rate in the U.S. Is higher in men and women from lung cancer. So the more people die of lung cancer, especially males, because more of them smoke cigarettes, then the rate is much higher. Next is breast in females, and prostate in males. Colorectal is the same in both, pancreatic, and then going down the line. So you can see that even when you have cancer, it's not a death sentence, and especially now, a lot of people are living past their surgeries and treatments for cancer. Cancer cells do have some characteristics that can be easily recognized. So a normal cell, an animal cell, usually spreads out on a petri plate or in a tissue, whereas cancer cells often round up, and they've lost a lot of their cytoskeletal elements, or some of their cytoskeleton has been disrupted. In culture, they also do not adhere as readily to the culture medium or the plate, so you can actually separate them, because the normal cells will stick to the plate and the cancer cells will come off of the plate, so you can kind of wash them off and purify the cancer cells. Cancer cells are not contact inhibited either, so normal cells will look like this. They have pretty much nuclei that look equivalent in most cases, and also the cells are contact inhibited, so they don't overgrow the plate. Cancer cells, on the other hand, have a variety of sizes. The nuclei can be of different shape, and you can have what are called micronuclei, tiny bits of the nucleus and DNA that's broken off and become a separate small compartment. And they're not contact inhibited, so they will actually start growing on top of one another. And if you let them grow enough, they'll grow over the edge of the plates. So here's a micrograph of normal fibroblasts growing in culture, and you can see those have nuclei that are all about the same size, and they, although they extend, they don't overgrow. That is, once they've contacted another cell, they stop growing. Whereas cancer cells look very different, one cell to another, and are not contact inhibited, so they keep growing, and they're much rounder in shape rather than stretching out on the plate. primarily because the cytoskeletal elements are not organized in cancer cells. As cancer cells grow, they start with usually a single mutation, and then they become more and more disorganized as time goes on with more and more mutations. So if you start here with normal epithelia, and then you have a mutation, this A here, later you'll have some dysplasia because you'll have in the center some of those cells developing a second mutation or a third mutation. And so you have this growth of cells around here that are fairly normal, except they have one mutation. Then in the middle, they have an additional mutation, and then a further mutation from there as they spread out, and then more and more mutations as time goes on. So they start to become more and more disorganized. The nuclei become misshapen and of different sizes, and then eventually you lose control altogether, so they invade other tissues and eventually metastasize. So going a little further, first of all, you have a carcinoma in situ here. That is, it hasn't broken through to other tissues. Then you get breakage through the basal lamina to invade other tissues, and then invading a capillary, so then these cells can move to different locations, invade other tissues such as the liver many times, and then set up a tumor there. So this is essentially a skin cancer that has moved into the liver. It's not a liver cancer per se. Cancer is often staged, or almost always staged. So you can have a carcinoma in situ here at stage 0. That is, it hasn't invaded any other tissues. Stage 1 is it starts to invade other tissues. In stage 2, then it's invading more layers of the wall of the colon, and it's attracting blood vessels to it. So it now has a richer supply of nutrients. Stage 3 is when it's gone all the way through all of the layers and invaded the fatty layers around the colon. And now it's starting to move at least some of the cells into the lymph nodes. And then stage 4 is it moves from the lymph nodes and spreads to other organs as well. And this is metastasis then. When it moves to the lymph nodes, that's called micrometastasis, in that it's locally moved to a different area. And these cancers can be followed progressively by looking at the mutation. So an initial mutation in some types of colon cancer is a mutation in the APC gene, which is a gene that controls part of the cell cycle, in that once this gene is and the protein that it forms is lost, then you get more and more cell division. Later on, you can have another gene. We've talked about the RAS gene before, the KRAS in this case. Then you get hyperproliferation of those cells. Eventually, a mutation in DCC causes even more cell proliferation. A loss of p53 means that cell those cells now become immortal. That is, the p53 controls apoptosis, and now that apoptosis does not occur, so the cells live forever essentially. And then other mutations occur, causing a carcinoma, and then you get invasion eventually and metastasis. Here's another example, more detailed, and this is in a pancreatic cancer. So in the normal tissue, there's essentially one mutation that sets off this progression, and that's in the mucin 1 gene. And then there's KRAS mutation and telomere shortening causing the KRAS to be mutated or essentially be destroyed. And then other mutations in other genes, the PSCA, mucin FASN, and then P16, mucin cyclin D, controlling the cell cycle, P53, which controls apoptosis, and BRCA2, which recognizes DNA damage topoisomerase 2, etc. And then, so you can see this progression, first cell proliferation, and then forming of more and more odd cells, odd-shaped cells, and odd nuclei, and eventually invasion and metastasis down here. And so this cancer has quite a low 5-year survival rate, down around 5%. So once you've gone down this line and finally people recognize that there's a tumor there, it rapidly metastasizes into other tissues as well. And here I've summarized mutations in 3 different types of cancers: colorectal, prostate, and pancreatic. And this is in just one of each of those types of cancers. So up here, you can also look at the karyotype, going from normal tissue all the way to metastasis down here. LOH means loss of heterozygosity, meaning that one of the chromosomes has lost that gene, and now you only have one copy of those particular genes. And the Q means that's on the long arm, and the P means it's on the short arm of those particular chromosomes. You also have gains, that is, duplications of certain regions of the chromosomes in some cases, listed here. And then there's methylation of some of the genes, which changes their expression, and then gene mutations, in this case starting with APC. You can find KRAS and P53 are common in some of the types of cancers, and then eventually BAX, which controls apoptosis as well. And then in prostate cancer, you have fewer overall chromosome changes, but many changes to several of the other regions, including, again, P53 and RAF, which is in line in that pathway that RAS controls as well. And then down here in pancreatic cancer, again, some of those same genes being mutated, including KRAS and P53, and in this case BRCA2, which recognizes DNA damage, and then mesothelin, which sort of loosens the cells up so that they can start metastasizing and moving around. And here are the categories here, if you want to look at each one of those. So you can see it involves genes for tumor suppression, for DNA repair, a growth regulator, signal transduction, transcription regulation, etc. So when you summarize all of those and look at what genes are mutated in cancer cells, you find that there's transcriptional regulation control is a large proportion, cell adhesion and motility. That is, they lose their ability to stay in one place and start metastasizing. Signal transduction, transport, intracellular trafficking, cell metabolism. So these are pretty common categories for cancer cells. So if you look at 2 very different types of cancers, breast cancers and colorectal cancers, you can see that the proportion in each one of those, cell adhesion and motility, signal transduction, transcription regulation, transport, are the same ones that you find in both of them. Even the ones that are more minor as far as being mutated in cancer cells, those are also in common to these 2 different types of cancers. And here is a diagram I think I showed early on when we were talking about signal transduction. So there are many different molecules and receptors, etc., that are involved in regulation of the cell and its interactions with the environment. So you have growth factors and cadherins, which are cell-to-cell contact proteins. There are receptors for those, and there are signal transducers such as RAS, and then regulators of signal transduction, regulators of transcription, and APC actually controls one of those. So there's a transcription regulator. If APC holds onto that, then it doesn't cause increased transcription. But if APC is mutated, then these catenins can actually turn on transcription quite heavily. Then there are many controllers in the nucleus, such as those that control the cell cycle, some that increase the cell cycle, DNA replication, etc. Some that recognize and accomplish parts of DNA repair, including BRCA1 and BRCA2. And then things that control apoptosis, such as p53, and then apoptosis inhibitors, such as BCL proteins. So here's just one example of that, and it's mainly the action of RAS. So you have a growth factor that sits on the growth factor receptor. If that attaches, that changes the conformation of this protein. And that signals this protein, which changes its conformation to activate RAS. And RAS is activated by dropping the GDP, picking up GTP, and then signaling other proteins such as RAF. So either RAS or RAF, if they are mutated, can cause cancers, initiation of cancers. Or you can block this also in mutant RAS for dropping the GTP and picking up GDP and activating it. But that signals the MAP kinase pathway, and that stands for the mitogen activated protein kinase pathway. So this actually starts phosphorylating certain proteins that activates them and activates transcription. So it then causes the cell to progress through the cell cycle and to replicate the DNA, and the cells start dividing then. So you get cell proliferation. So you can see in a mutant RAS, it will continually be activated because it cannot drop the GTP. And whether or not you have a growth factor out here stimulating or not, it continues to be activated and increase transcription and cell division. And here are a few more details on that. So there's the receptor for the hormone, the growth factor, that then signals other proteins inside the cell, which causes RAS to pick up the GTP. RAF is coming from a different pathway and being activated by RAS to initiate the MAP kinase pathway that then phosphorylates a number of transcription factors, including JUN and MYC, which are two very prevalent ones that are found turned on in cancer cells. And here is a more detailed view of that. And all of the labels in red, those are all proteins and also sometimes hormones that when they're mutated can cause a cancer, or at least they've been found mutated in cancer cells. So that includes RAS here, as well as NFK, SRC, ABL for Abelson. There's JUN and MYC, those are transcription factors. Here's BCL2, which is an inhibitor of apoptosis. And over here is p53, which signals apoptosis, so that stimulates apoptosis. So many different proteins in here along the way are common in various types of cancers. And here I've just separated these by category. So here you have the ligands, the molecules that attach to the receptors, and there are the receptors that are known in different types of cancers, and then signal transduction in here, and then gene expression control, cell cycle control, DNA repair, and apoptosis. So each one of those categories are types of proteins, genes, and then proteins that when mutated have been found in various types of cancer cells. And here is even more detail about some of those pathways, showing that many different proteins are involved in these pathways that essentially take a signal from the outside and then transfer that signal to the inside. And then there's all of this signal transduction inside the cell that's highly controlled, so that in the end, this will control when and where those genes will be transcribed and expressed in these cells. And here's another different view of that, showing some of these genes and proteins again. And this is when you have damaged DNA. What are the signals that then cause the cell to go through apoptosis? And one of those is BRCA1 here, which senses some of those mutations, and then sends a signal down here either to repair the DNA or to send a signal over here to p53 to start the process of apoptosis. And here's a more concise figure of something similar. So BRCA1 is a very central part of this whole process. So it recognizes when there is DNA damage up here, and then it actually controls many processes, including X chromosome silencing, non-homologous end joining of DNA, signaling repair functions, transcriptional regulation, homologous recombination, and other parts of DNA repair and checkpoint regulation of the cell cycle, that is, proceeding from G2 to mitosis, also from G1 to the S phase. So it's central to this. And BRCA2 is also involved, but only to a smaller extent. So mutations in BRCA1 and BRCA2 often lead to breast cancers, also to ovarian cancers, and some other types of cancers as well. Here's a diagram showing how some of this relates to an embryonic or a developmental stage that the cancer cells revert to. And this is one that is in for that APC gene of the indicated in colon cancer. And APC stands for adenomatous polyposis coli. And this occurs in families where you have a high number of polyps in the colon, that many of which develop into cancers. So in the fetus, when the colon is being built up, you need increased cell division and growth. And so all of these pathways, including the RAS pathway here that increases the MAP kinase and increase in transcription. And then you have beta-catenins that also increase transcription. And then over here, the APC controls somewhat the phosphorylation of these proteins over here. Now, in an adult, then the APC acts on those different molecules to break down the beta-catenin. So it signals phosphorylation of beta-catenin, and then that degrades the beta-catenin that actually beta-catenin itself will increase transcription. So this stops transcription and essentially cell division and cell proliferation. However, when the APC is mutated, it cannot act on those different proteins here and phosphorylate the beta-catenin. So the beta-catenin is still produced in high amounts. And that increases transcription in the nucleus and increases cell division and growth, forming those polyps and eventually tumors. Okay, so that's the end of part 1 on cancer, talking about some of the cellular aspects and a bit of the molecular, some of the molecular aspects. The next video, part 2, will focus more on the molecular aspects of tumorigenesis and carcinogenesis. Okay, so I will talk to you later then.